All right, I want to just make sure to take a second to go over everything from the Gizmo in part B so that you kind of understand it a little bit better because it is different than the way that we learned the information on genetics, and that is because they want to be able to show you what's going on. So I don't want you to think of these necessarily as codons because we know that codons are groups of three nucleotides that each code for a particular amino acid. So instead, what we're going to do is look at these as like three chunks of genetic information that code for a trait. Okay. So when we're looking at and analyzing the DNA sequences, think of either, um, a gene is, or a part of a gene is present or isn't, but it's going to be the differences between them that really determine whether or not one allele, one version of the gene, or a different allele version of the gene is going to be expressed. So go ahead and read the introduction. And I mean it, make sure to read it. So go ahead and pause this video so that you can go through and read the introduction. Okay, so what you need to do is you're going through the questions here. You can do parts 1 and 2, but I want to highlight what's going on with part number 3 because this gets a little bit tricky. So when we are looking at, you already did the twins, when you're looking at the populations, you're going to need to find the DNA sequence and then you need to compare it. So let's say, you know what, I'm not going to do one that you need to do. Let's start with this little froggy back here. Here's froggy I, and I'm going to scan their genome and pull this up here. So every one of these little blocks is going to be some piece of a gene. And so all of them together make up the gene and code for some kind of trait. Now, if I want to compare, what did I have? Froggy I with, let's go polar opposites here and go with Froggy P. And I take a look at Froggy P, it's really hard to see what is in common and what's different between them, especially since I have to be reading it in groups of 3. So what we need to make sure to do is turn on the comparison guide. That comparison guide is going to highlight 3 at a time, so it's a lot easier to see what's happening. And so we see here that these two are not the same. If I scan down 1, I can maybe compare that, but don't do that. So the genes, the genetic information here, what they're calling codons, is set up to be in groups of 3. So we can't just start on any piece of information. We have to make sure that we are keeping them all in groups of 3. So if I look over here on the side, I can see that I am currently highlighting nucleotides 1 to 3 to be able to make sure that I am staying into groups of 3. We always want that lower number to be a multiple of 3. So I went from 1 to 3. Now I'm looking at 4 to 6. Those are different. Now I'm looking at 7 to 9. I see that those are different. 10 to 12, etc. And just like it says in the instructions, 15, you are not going to have a full one at the end. There's just part of one. I don't know why they did that. Seemed like a good idea at the time, I'm sure. But anyway, as you're going through, then you have right here, it says scan the frogs, blah, blah, blah. And you're going to insert a screenshot of that. And as a reminder, what you're screenshotting is going to be just this information right here with the appropriate part of the comparison guide shown. And to do your screenshot, you're going to hit Command Shift 4. Let me make sure that's true. Yeah. And then you're going to get this little symbol. And so you can go ahead and highlight whatever it is that you want to screenshot. And then you can just drag the image that comes onto your desktop directly into the document so that that is there. So if you've got questions, email me or I'll stay on Zoom, but I want to make sure that you have the information you need when you need it. Have a great day, and I will see you guys tomorrow. Don't forget about the quiz.